Hello viewer, welcome to the YouTube channel FahadYounas. Today I will discuss the topic the discovery and structure of bacteria. Introduction to bacteria. Bacteria are single celled microorganisms that can be found in a variety of environments. They have a simple cell structure and can reproduce quickly. This discovery of bacteria has revolutionized the field of microbiology and has led to many important scientific breakthroughs. Discovery of bacteria. Antonie van Leeuwenhoek discovered bacteria in 1676 using his own microscope. Bacteria are unicellular organisms that can be harmful or beneficial to humans. Types of bacteria. Bacteria are single celled microorganisms that can be found almost everywhere. Some bacteria are harmful while others are useful and necessary for life. There are three main types of bacteria: spherical, rod shaped, and spiral shaped bacteria. Structure of bacteria. Bacteria are single celled organisms that can be found almost everywhere on Earth. They have a cell membrane, cytoplasm, and a nucleid containing the genetic material. Some bacteria also have external structures like flagella or pili that them move on attached to surface. Importance of bacteria. Bacteria are single celled microorganisms that play a vital role in many aspects of life on Earth, including human health and the environment. They have a simple but unique structure and can be found almost everywhere. Discovery of bacteria. The discovery and understanding of bacteria is a fascinating and important chapter in the history of microbiology. Bacteria are single celled microorganisms that are among the most abundant life form on Earth. They play critical roles in various ecosystems from aiding in digestion to causing diseases. Here's an overview on the discovery and understanding of bacteria. Antonie van Leeuwenhoek, 1676. This discovery of bacteria began with the invention of the microscope in the late 17th century. Dutch scientist Antonie van Leeuwenhoek, using a simple single lens microscope he designed, became the first person to observe and document microorganisms. He called these tiny living creatures animalcules, although he didn't understanding their significance. His observation marked the beginning of microbiology. Lewis Pasteur, 19th century. In the mid 19th century, the field of microbiology started to advance rapidly. Lewis Pasteur, a French chemist and microbiologist, made significant contribution. He conducted experiments that disproved the theory of spontaneous generation, which proposed that life could arise spontaneously from non-living matter. His work showed that microorganisms, including bacteria, were responsible for fermentation and the spoilage of food and beverages. Robert Koch, late 19th century German physician and bacteriologist, Robert Koch made pioneering contributions to understanding the role of specific bacteria in causing diseases. Koch postulates a set of criteria became the gold standard for establishing a causative relationship between a particular microorganism and a specific disease. Gram staining, 1884. Danish bacteriologist Heinz Christian Gram developed the Gram staining technique, which allows for the classification of bacteria into two major groups: gram positive and gram negative, based on the characteristics on their cell wall. This technique is still widely used in microorganisms today. Structure understanding as microscopes improved, scientists were able to study the structures of bacteria more closely. They found that bacteria have a variety of shapes, spheres, rods, and spirals, etc. The possess complex structures, including cell wall, flagella, and capsule. This understanding of bacteria structure contributed to their classification and identification. Genetic understanding. In the 20th century, advances in genetic and molecular biology further depending over understanding of bacteria. This discovery of DNA roles in the heredity and the development of techniques like polymers, chain reaction, PCR, allowed scientists to study bacterial genetic genes expression and DNA sequences. Microbiome research. In recent years, research on the human microbiome has gained prominence. The human microbiome is the community of microbiology, including bacteria that habitat the human body. These studies have relieved the essential role that bacteria role in human health, from aiding digestion to influencing the immune system. Understanding bacteria has had a profound impact on medicine, agricultural, biotechnology, and various other fields. It has led to the development of antibiotics, vaccines, and a deeper knowledge of ecosystem and the role of microorganisms within them. Bacteria remain a vital area of scientific research and continue to shape our understanding of life on Earth. Structure of bacteria. Bacteria are single called microorganism within a simple but highly organized structure. Their structure can vary depending on the bacterial shapes, but there are some common features. Cell wall. Most bacteria have a rigid cell wall that provides structural support and protection. The composition of the cell wall can vary. There are two major categories of bacterial cells walls based on their response to gram staining: gram positive bacteria and gram negative bacteria. Gram positive bacteria, these bacteria has thick peptidoglycan layer in their cell wall which retains the crystal violet stain used in gram staining. They may also have tachyoic acids and lipotachyoic acids. Gram negative bacteria. These bacteria have a thinner peptidoglycan layer surrounded by an outer membrane which contains liposaccharides. Gram negative bacteria do not retain the crystal violet stain and take up the content stains in gram staining. Cell membrane, plasma membrane. This cell membrane is located just beneath the cell wall. It's where as a selective barrier controlling the movement of substances in and out of the cell. It is made up of a phospholipid bilayer and contain various proteins, including transport proteins and enzymes. Cytoplasm. The cytoplasm is a gel-like substance inside the bacterial cell where most cellular activities occur. It contains enzyme ribosomes, nucleide, and various inclusions. Nucleide. Bacterial cells do not have a true nucleus but instead have a region called a nucleide where the bacterial chromosome, a single cellular DNA molecule, is located. This DNA carries genetic information for the cell. Ribosome. Bacterial ribosomes are involved in protein synthesis. They are smaller than eukaryotic ribosomes and are classified as 17th ribosomes, consisting of 50s and 30s subunits. Flagella. Some bacteria have flagella, which are whip-like structures used for locomotion. Flagella can be present on the cell surface and vary in number and arrangement. Believe these are short, hair like appendages on the surface of bacterial cells. They play various roles, including additions to surfaces to other cells. Capsule. Capsule. Some bacteria have a protective capsule outside the cell wall. This capsule is composed of polysaccharides and help protect the cell from the host immune system and dissection. Endospores. Certain bacteria can form endospores, which are highly resistant dormant structure that can withstand extreme environmental conditions. Endospores are produced by a few bacterial genera as a survival mechanism. Plasmid. Plasmid. Bacteria makes or contain plasmid, small circular DNA molecules that carry genes for specific functions. Plasmid can be transferred between bacteria and play a role in antibiotic resistance and other genetic adaptations. It's important to note that the structure of bacteria can vary widely among different species, and some bacteria may have additional structure and adaptation specific to their ecological niche and lifestyle. The study of bacterial structure is the fundamental aspect of microbiology and has contributed to our understanding of bacterial function, diversity, and pathogenicity. Thank you for joining me on this journey through the discovery and structure of bacteria. We appreciate your support for FahadYounas channel. Please consider sharing, liking, and subscribing to our channel. Thank you for watching my video and share.